Good morning, everyone. Welcome to the Fourth Quantu Project Workshop. It's my pleasure to be hosting you this morning. We're going to be talking about how daily activities like sleep and exercise affect brain health. With me is Jenny Brethen, the Quantu Project Manager, Dr. Byron Long, a computer scientist, and somebody who helped us start the study 3 years ago in Houston. Erin Pole, who's a graduate student in the lab and a sleep specialist. She's studying sleep algorithms to interpret patterns of sleep as a function of daily activity and how it affects brain health. Zach Maloney, who's a second-year graduate student in biomedical engineering as well, and he's focused on the neurogenesis or brain in a dish assays for this project. Ash, Ali, and Thomason are our undergraduate researchers who are helping on this project. And of course, we have our volunteers and friends who are joining us on this call. So thank everyone for joining us for this fourth Quantu Project workshop. I'm going to briefly go over the workshop details and the agenda. I'm going to start by sharing the background on what is known in the literature about how exercise, sleep, and other daily activities change our brains. Then Erin's going to pick up and describe how we're studying sleep patterns in individuals for this project, and also give an update on what we're identifying as unique fitness patterns during the pandemic. Zach's going to describe some of our neurogenesis assays and our brain in a dish assays to study the area of the brain regulating sleep, and then also describe briefly what we're starting to do in terms of retina imaging for looking at longitudinal effects on the neurovasculature from daily activities and things like even infections. Then Jenny and Byron are going to follow up with describing our cognitive testing, the brain health intervention studies, and how we can have registration renewals for volunteers. Then we're going to open it up for questions. As a brief background to how this project started, I was inspired by the woman you're seeing in this picture here, Marian Diamond. She was my neuroanatomy professor at Berkeley when I was a graduate student there, and she would enter the classroom with this giant hat box, beautiful, elaborate hat box, which she would open up, and inside that hat box was a human brain, just like she's holding in this picture. And she would point to different regions of the brain based off of what we were studying in that particular class, and describe the function and the structure of the region of the brain of interest. She's also famous for a number of other things. She was sent Einstein's brain after he died, parts of his brain, and she identified that he had more astrocytes or neuron helper cells, glia, 2 neurons. Their ratio was higher than normal average male brain in certain regions of the brain. She also is famous for identifying ways that brains can maintain their health across a lifespan. And she did it in animal models. And she summarized her work this way in colloquial language when she wrote her autobiography in the Journal for the Society of Neuroscience. She summarized it by saying healthy brains really need good diet, exercise, challenge, newness, and love. And for her, in the mouse brain she was studying, the rodent brain she was studying, diet meant the normal chow versus the high-fat diet. Exercise was the animals were allowed to regulate exercise and run on their exercise wheels. Challenge and newness meant the animals were exposed to new things in their environments, in their cages, so that they were always constantly learning and improving in a way, the way that they were thinking. And in terms of love, she was looking at social bonding metrics and neurochemicals involved in that process, like oxytocin and other things that might improve the brain related to social activity. What I want to highlight is that while she was studying a mouse model, other models of the brain have also been studied in terms of what helps improve cognition. You can see pictures of all the different types of models and model systems, animal models that are being studied in research right now, used in research right now. You can see they have different shapes and different structures and sizes, of course. And when you compare the mouse brain, which Marian Diamond was focused on, to the human brain, which we're focused on for the study, you can see that there's vast differences in structures, and of course size, and of course we know function. So our goal for this project is to really take what's been known in the literature and what's been studied in animal models and what's known about human brains to understand how daily activity can be used to protect and even enhance cognition. What I'm showing you here is a summary of the ways that cognition can decline. So the molecular and cellular processes which are associated with age-associated cognitive impairment or dementia are shown on the left of the green highlighted box, or where the symptoms of cognitive change show up. So in the 6 boxes on the left, you'll see examples of ways that the brain can decline. Inflammation and viruses are associated with cognitive impairment later in life. Disrupted proteostasis. Many of you may have heard about A beta proteins or tau proteins affiliated with Alzheimer's disease. Their abnormal accumulation into associated aggregates of proteins have been affiliated with inflammation and symptoms of Alzheimer's disease. Aberrant metabolism has as well. So some people describe Alzheimer's as diabetes 3. Of course, DNA damage and brain cell death leads to cognitive impairment, can lead to cognitive impairment. And the brain lymphatics, or your clearing system for your brain, and the microvasculature, which is critical for bringing in nutrients and taking out toxins, also can be damaged and affect your cognition. What we're interested in is something that's common across all of these ways that you can see decline. And it's also a way we believe that can be used to harness and improve cognition over time. And that's the way the brain repairs itself. So in human adults, there's limited ability of the brain to self-repair and regenerate. We're focused on animal models and experimental models of human systems in the lab to understand how brains regenerate and actually to revert some of the symptoms of cognitive impairment and understand how the cellular changes are happening and whether or not there's new cells forming and whether or not there's brain tissue that can be regenerated as a function of daily activity. So that's what this project is about, the Quantu project. We also are harnessing a lot of existing literature and understanding how daily activities affect the brain. Most of this, the vast majority of this, not nearly all of the big studies, have been done qualitatively, meaning they're not using trackers or teasing out detailed daily activity for individuals, but they're making really salient and critical points about how general habits protect the brain. For instance, many of us may be familiar with knowing that a high-quality diet improves the human brain. So things like lowering your sugar intake or keeping it low and limiting red meat, limiting the much of alcohol you have, and increasing the amount of mixed vegetables and whole fruits, grains and legumes, olive oil and nuts you have. Interacting, a social activity, of course, are critical for human brains, as are exercise, keeping up hygiene, limiting exposure to viruses, which is critical right now, and doing mentally stimulating activities, learning, being exposed to new things. All of these have a background of many different research projects and experiments that have shown in humans that these are protective activities for the brain. If we look at this more closely, you can see that this has to be continuous. It's not a one-off. You can't just do it one day. But a 2-year combination therapy of these behavioral interventions can protect cognitive functions. And if you look at it in more detail, it can literally change cognitive scores if you adopt a healthy diet on a daily basis. As this example article shows in a retrospective study of over 2,000 people. What also is really exciting for us in this project is that even if you have a genetic predisposition to cognitive impairment, for example, if you have a variant of ApoE associated with Alzheimer's disease, you can change your outcome, meaning you can improve your cognition based off of adopting a healthy lifestyle. That's what this project is really trying to figure out. How do individuals respond to different lifestyles and improve their cognition so that they're maximizing their cognitive health across a lifespan? One of the ways we think we can do it is through optimizing high quality sleep. And sleep is really interesting because it's one behavior that's little studied and yet so critical for the health of the brain. So during one night of sleep, we know that your brain has different circulating compounds that are changing your metabolism in your brain. You're clearing out toxins, you're changing your blood flow, you're changing your cerebral spinal fluid flow throughout your brain as you're sleeping. One other thing that's known is that if you have one night of sleep deprivation, and this is in a healthy human, you have more accumulation of A beta proteins, those proteins associated with Alzheimer's, in the cerebral spinal fluid and in the blood. And if you can look at this longitudinally, if sleep is disrupted across the lifespan, we know it's been associated with and even predictive of cognitive impairment or dementia. This particular type of sleep that's been associated with it, one of many, is rapid eye movement sleep. So rapid eye movement sleep is a light sleep that Erin's going to talk a little bit more about later in this workshop. It allows the brain to self-repair in terms of synapses reforming or changing. It also is involved in memory formation, and it can be involved in other processes of brain repair. So we're interested in understanding this in detail for individuals enrolled in our study. One of the reasons is because we think also that sleep is regulating neurogenesis, and REM sleep might be associated with it as well. So in the past, about 4 decades ago, it was thought that whatever brain cells we were born with, we died with, meaning we couldn't form new brain cells. But in the past couple of years, past decade even, there's been a paradigm shift. We know neurogenesis happens in the adult human brain, even in the hippocampus. So when you were in your teens and when you were in 20s and 30s, you could have new neurons form every day. But in your 60s and 70s, you can as well, same amount. This study in particular showed that everyone throughout their healthy lifespan has an ability to form new brain cells on a daily basis, on a regular basis. But those people who have Alzheimer's disease or cognitive impairment have changes to the ability to form new brain cells. One of the exciting other very recent findings, in fact, of July of this year, was that there's markers in the blood after exercise that are associated with new brain cell formation. This is a study that follows up on other experiments that have been also shown in animal models. And this particular study identified a compound, GPLD1, which is promoting new brain cells that is after exercise. And what they did is actually they studied exercise in old mice and exercise in young mice, and they took the blood from these mice and they gave the blood to sedentary older mice and found that regardless of whether it was an old mouse it came from or a young mouse, if that mouse had been exercising and improved cognitive tasks that the mouse did in the sedentary older mouse when they were transferred or had a transfusion of the blood from the exercising mice. So that's really promising for the work we're doing in the lab because we're looking at neurogenesis from cells that are derived from our volunteers. This is a snapshot of a brain organoid. You're seeing in blue neural stem cells and neurons. And those are the nuclei that are stained in blue. Green are stem cells. These are markers of nestin, or a protein which is associated with stem cells. If we zoom in, we're interested in the cells which are purple here. These cells are proliferating. They're stained for Ki67, a proliferation marker. We're looking at where are these cells forming across the development of these cortical organoids or particular regions of the brain which are regulating sleep, which Zach will present a little bit more about. If we look at another snapshot we have from the lab, we're looking at many different markers of brain cells, not just neurons, which in this particular image are shown in green, stained for their neural filament proteins. We're also looking at astrocytes or glia, the ones that I mentioned were high in Einstein's brain in certain regions. And we're also looking at specific proteins which are expressed in healthy brains, and those that are different in brains derived from those who have genetic predisposition to Alzheimer's and those who are changing their activity patterns. So we're looking at these in 2 and 3D. These neural networks that we're forming in the lab allow us to distinguish different features of how neurons form from humans. And we've looked at it for different diseases already. We've shown in a neural developmental disorder for children called Smith-Lemli-Opitz syndrome that the networks form differently than they do in healthy control cells that are derived from healthy individuals. So we're following up with this for the Quantu project study for our volunteers who are also being tracked longitudinally for their changes in activity. So the goal of this project overall is to link what we're able to study in the lab in a healthy way of looking at brain cells to what we can measure in humans in real time, in real life, using wearable devices. So we're looking at capturing sleep over the course of many months to years, and the patterns in sleep as a function of what we can measure from wearable devices, and then correlating it or even potentially making it a prediction of or predicted by changes we can observe at the protein level or at the cell level in terms of these functional neural networks in the lab. As a summary of where we are, we started 3 years ago back in Houston when I started actually with a cohort of students who were willing guinea pigs to be able to tease out different patterns of their own sleep and social activities. We opened up in 2017 to recruit volunteers who were healthy between the ages of 18 to 32, who started to wear wearable devices and be tracked in terms of their sleep and their daily activity. We started also looking at blood samples in combination with looking at cognitive test scores, and what we're doing now, introducing intervention studies. So right now we're in the full study. We're recruiting volunteers and children as young as 2 years old, all the way up to adults who we have in their late 80s and 90s. And for this, we're having a subset of our volunteers have bioassays and intervention studies and cognitive tests, proteomics and epigenetic screening, and genetic sequencing. And all of our volunteers are having daily tracking. So people can sign up to be enrolled as a full volunteer, or they can sign up just to be part of our tracking study. For those who are just doing tracking, we encourage you to use your own wearable devices. And we're opening up to anyone who wants to join, who has their own wearable devices. But for the full study volunteers, we provide the devices, and we also provide all of these experimental bioassays and other medical exams and clinical exams. It's a research study, so we're not providing medical advice. We're learning from this information to be able to make a difference and impact in brain health for society in general and for everyone broadly whose families are affected by different neurological disorders. We are recruiting across the board for different cohorts. So we've been recruiting predominantly for control healthy individuals across different ages. We're also recruiting for healthy athletes. So we've recruited CrossFit athletes who are elite competitive athletes. We've also recruiting runners. We also have people who are diagnosed with sleep disorders and those who have mild cognitive impairment and Alzheimer's disease in the study. We're recruiting across the nation and even internationally, although we have our main sites in San Antonio here, in Houston, Texas, Los Angeles, California, and the San Francisco Bay Area. With that, I'd like to just summarize what we're starting to do with our intervention studies. We're having a number of our volunteers who are interested sign up for sleep hygiene interventions, cardiovascular training, or learning a new language. For our first intervention, we're offering a Python course where people can learn to code in Python. That's not something you've already been exposed to. And then before and after that intervention, we'll be taking blood draws to see if there's changes in proteins, and in studying in the bioassays, the functional assays in the lab. We'll also be having parallel cognitive test scores and the retina imaging. With that, I'd like to start to turn it over to Erin Pollard.